First speaker will be Eckhard Wimmer from SUNY Stony Brook, who's going to be telling us about whole viral genome synthesis and synthetic biology.
Thank you very much for giving me a chance to speak here. I love the meeting. It's a great place and great talk so far. As you heard, I will talk about whole viral genome synthesis and synthetic biology with an emphasis actually on probably a little bit on vaccine production. And the people who are listed here are the people who are in the laboratory. I would like to point out two individuals, particularly Dimitris Pepamichail, who was a graduate student in Steve Skina's laboratory. Steve is a computer scientist and in the Department of Computer Science. He's on sabbatical here in Hong Kong and available for you to ask questions that I undoubtedly can't answer. So the system we're talking about is this one here. That's the empirical formula of poliovirus that we synthesized in the year 2002. And if you put the atoms into order, then you get a beautiful particle, an icosahedron, naked, no membranous shell with a 5-fold, a 3-fold, and a 2-fold axis. And for reasons I don't want to explain right now, don't have time to, we synthesized this, and I'm showing you the scheme that we used, because it's an old hat, putting together oligonucleotides until we had the DNA. Then we transcribed the DNA into RNA with T7 RNA polymerase. And then we did something that actually nobody can reproduce with any other virus systems yet. You take the RNA and put them into a cell-free extract, and in that cell-free extract, the virus will grow and will eventually make particles. And this actually cell-free synthesis of poliovirus outside living cells is still a unique property. It was totally ignored in 2002. In fact, we got beaten up for this work, even though I loved the work, and I still do. Beaten up for several reasons, not by everybody, I should say. And the emotions slowly, but only slowly, have cooled down. Now, is a list of genomes, of viral genomes that have been synthesized since. It started with poliovirus, then came the work of Ham Smith with phage 5X174. It's a very beautiful piece of work where all the 5,386 nucleotides could be put together in 2 weeks. I was truly jealous when I read the paper. It was great. At the same time, there was a paper by Andy Drew's group where they refactored actually part of phage T7 to their own plan, how for their own purpose the genome of phage T7 should look like, and they synthesized for that 11,550 nucleotides. And then in 2005, we had the synthesis of the 1980 influenza virus by Tampi and his colleagues, which was, of course, a sensation. And in fact, had it been published not in 2005, but in 2002, and had it been the first viral synthesis, there would have been much more of an uproar than we experienced in 2002. We will here talk about that and the importance of that, and in fact, the way it was embedded into more or less calming documentaries, saying, don't be worried, it's important and necessary that we synthesized the virus and we published every detail of it. Now, a little later, there was the synthesis of 2 endogenous human retroviruses. It's a very interesting story. You know that we all have a lot of genome sequences in form of endogenous viruses. In fact, these viruses are symbionts. With other words, they're necessary and very useful for us. And if we wouldn't have them, I think women couldn't have a baby. Not very many people know about that. My computer doesn't like this here. But anybody help me to get my pictures up there again? larger. Okay. Well, anyway, these retroviruses are symbionts. In other words, they co-evolve with us for better part of 5 million people. And they're necessary because their gene products allow the woman to build a proper placenta. And then in 2007, there was the synthesis of the SIV from chimpanzees, which finally gave the ultimate proof that HIV evolved from the chimpanzees' progenitor trains. And this year, just the largest RNA virus was synthesized by Ralph, whom we will hear right after my talk. Now here are the genomes again in a different order, from smaller to bigger. And you'll see that the largest genome of an organism, although it hasn't been booted to life yet, is that of Mycoplasma genitalium, and we heard about it yesterday. Fantastic piece of work, and it's hope that it will be booted to life soon. And if you look at this here, then you ask the question, this is a life, of course, entity, as it's been called. It's a bacterium, and how about viruses? Well, viruses are usually considered to be dead in a way. And if you ask, actually, if you scan people, do you think a virus is alive or is dead? It's being considered dead. But this is only in the circles of scientists. In fact, when we published our paper on the poliovirus synthesis, we got rather nasty email messages telling us that we wanted to play God, that we tried to create life in the test tube, that we wanted to be little Frankensteins, where the little was actually an insult. But the point is that in synthetic biology, we have to begin to think about how to define life in a different way and how to formulate it. You remember probably all that when the synthesis of this very large genome was published, it said in front pages, Craig Venter synthesized life in the test tube. And of course, that's scary for a lot of people. And I think as we will modify organisms, they will be misunderstood as new life forms, when in fact they're misunderstood as new life forms that are different, fundamentally different from what existed before. And we have to actually define this better, or else there will be a lot of uproar and tension in the society. And I think this is a meeting where this should be sort of discussed. When you think about it, that here's a virus whose genome is twice as large as that of Mycoplasma genitalium, then you ask yourself, why would we call this mimivirus dead and this organism alive? Compare that with the genome here of those endosymbionts, very small bacteria, if you want to, 160,000 base pairs, smaller than actually the genome of smallpox virus. So eventually, I think the division of what is alive and what is not alive doesn't no longer make sense. And when we change actually living organisms or organisms that we consider alive to doing things that are commercially advantageous to us, we should avoid calling them new life forms, because in fact every mutation that we made in poliovirus or in yeast or in others could be called a new life form. And I think that's a problem that I think we should watch out for. Now, the synthesis of poliovirus you couldn't really put in the category of synthetic biology, because we didn't really change much of this virus except for a few watermarks. But we have recently looked at the degeneracy of the genome, of the genetic code, and putting a different codon usage and codon pair usage into the poliovirus genome in order to ask what the effect would be. In fact, there are two possibilities to change that, to change the codon for codon pair usage in a viral genome. One is to change the synonymous codon use, which actually then will lead to a codon bias in codon use, as you're all aware of that. The codons used in E. coli is not the same as codons used in human. And then the other one, which is actually scientifically more interesting, the synonymous codon pair bias. And that's what I will mainly talk about it. Stefan Müller 2 years ago talked about the synonymous codon use and the changes that we've made in order to study that in a replicating system like poliovirus, which we were very comfortable with. And you have codons that are frequently used and codons which are very infrequently used. Now here is, I think I switched one too much. Here is the codon use of the poliovirus, a segment of the poliovirus genome. And that just shows you the different kinds of codons used for each amino acid in what percentage. In this case, we just shoveled these, but we didn't change any codons. We randomized them. We call that poliovirus SD. Whereas here, in this case, we used rarely used codons for this viral gene segment. For example, this codon here was almost completely replaced from, these codons were completely replaced to this rare codon here. And then we put a segment, synthetic segment, into what normally encodes the capsid of the viral genome. This is the front part of a polyprotein that the virus makes. It's one large protein that by proteolytic processing, you make then smaller proteins. Eventually, you assemble the virus. If you did something wrong with the coding sequence here, there will be an effect on all of these proteins because the ribosome goes from left to right. And therefore, if there's a poor translation here, these replication proteins will be also fully replicated. And so when we made actually a virus with predominantly rare codons, it was not very surprising that this virus was not viable. However, it was interesting that this virus, where we just shoveled the genomes without, shoveled the codons in that segment without changing codons, that this virus was replicating with normal replication kinetics in spite of having 943 silent mutations. Which means at least in that segment of this replicating entity, the introduction of numerous mutations didn't make a difference. Now let's go then to codon pair bias. In codon bias, we're changing codons. In codon pair bias, we do not change codons. Let's look at this hexapeptide here with that sequence. If we change codon pairs here, that is to say, if we take one of the amino acids, one of the codons from one switch here to another and switch it over to here, none of the codons have changed, but their array in the genome have changed. And there are three interesting properties of codon pair bias, and that's the following. If you have two codons, let's say GGG and CCC, that may give you a specific bias within the genome. But if you add two more codons in between here, the bias that you had here is no longer functioning. That is to say, their bias exists only between adjacent pairs, which is maybe not so surprising, but it's been first observed by Wes Hatfield, whose group actually discovered codon pair bias in E. coli. And third, actually, that pairs with the central CG or UA are highly underrepresented. So if you have a C here and a G here, that is a codon pair that the system simply doesn't like. And it's been actually proposed that codon pairs with such a CG, a C in 3 here and a G in 5, in 3 and a G in 1, position 1 of the next codons, are normally highly underrepresented, and the major reason for the codon pair bias. So how did we go about it? We looked at the human genome and calculated what the expected codon pairs would be. The statistically predicted frequency in the human genome at which a given amino acid pair is encoded by each possible synonymous codon pair. And then we compared that to the observed codon pairs, the actual genome-wide frequency of each codon pair. And if you divide one of the other, you get what we call the codon pair score. And if you then add up each score in a gene and divide it and make an average of them, you get the codon pair bias, or CPB. And that Dmitry Paha-Mikhail did for the human genome. Here is an example again for this hexapeptide. The codon pair score would be 0.2 here, minus 0.8, minus 0.5, and so on. And if you add these up, divide by 5, you come to a number of minus 2.8. And if you do this now for all annotated human genes that have been published, you get a curve here of human genes that centers at around plus 0.08, let's say. That is, everything that is above 0 are codon pairs which are more represented than expected, whereas anything below 0, these are codon pairs that are getting less, the frequency of which is less than expected. Poliovirus falls in this particular scheme to this position here, just slightly the point of 0. And so what we did is, in fact, we constructed a virus with the most overpresented codon pairs, which we call PV max, falls into this position here, or with the underrepresented codon pairs, which we call PV minus. Now this one has 566 silent mutations. This has 631 silent mutations. Now it's always important to realize that the codon usage and the amino acid sequences of these viruses that we constructed is constant. So in these experiments, the amino acid sequence of the virus is not changed whatsoever, and nor are the codons. The codons are just differently placed. Now, it would be difficult to do that. In fact, then these constructs are again are either codon pair with overrepresented or underrepresented in this particular segment here. We're comfortable with the segment because we know from work for years that in the RNA encoding that capsid region here, there are no RNA signals that are important for viral replication. If there were RNA signals, then of course changing the sequence would be detrimental. And of course, changing the codon bias actually for that, in fact, changes in the segment was enough to kill the virus. Now, if we take the segment and look at how many possible synonymous encodings for that region there would be 7.9 times 10 to the 442 possible synonymous encodings, which we're told is more than atoms in the known universe. This is actually very interesting. The question is, how did evolution arrive out of this huge number of possibilities at the encoding of the poliovirus genome, which changes, of course, from generation to generation of replication, because viruses are quasi-species, but in principle remains constant. Now, in order to select for poliovirus max or poliovirus min, we're using an algorithm which maximizes the codon pair bias either towards a negative or positive optimum. It retains the exact amino acid sequence, it retains exact synonymous codon usage, and it controls the RNA secondary structure of the target messenger RNA, so that you're sure that you wouldn't have, by chance, put into the RNA a hairpin that would interfere with replication. And the surprise was that if you put into the segment here underrepresented codon pairs, the virus would be dead. That is to say, translation going through here would be so hindered that the virus would not reach a minimum threshold for proliferation. And I will show you the data that the effect is on translation. Whereas if you put represented genomes here, the virus will grow very happily. Viability and the titers are almost like wild type. Now that was a surprise too, because it was actually concluded from West Health Field studies that if you let a ribosome going too fast through an open breeding frame, the growing change of the polypeptide would come out too fast. It wouldn't have the time to fold properly, and you would need pausing. And it was predicted that if you take overrepresented codon pairs, that we would actually find a virus that would be debilitated in replication. This was not the case. Now, the question was whether, in this case here of a dead virus, we hit hotspots. In other words, is there in this PV min a region? Is there in the PV min a region that, because of the changes of the sequence, it rendered the virus lethal? And so we subcloned several segments here into the X, Y, and Z region. And to cut the story short, if you combine more than two regions, the plot size actually gets very small, but the virus will still replicate. If you go to 3, the virus, of course, is dead. But all individually, none of these segments actually kill the virus. So there was no hotspot that we touched that would interfere with the virus. It's an additive debilitation of the virus. This virus died here because of the death of cut of thousands, or death by 1,000 cuts. There was another very interesting phenomenon and unexpected. Normally, poliovirus has a specific infectivity of 1 to 100 in HeLa cells. In other words, you need 100 particles in order to get one infectious event. 100 particles for one plaque-forming unit. And the Sabin strain, for example, that's been isolated from Albert Sabin, has about the same ratio, 1 to 200. This virus here, PB min Z, with one of the segments at the C-terminus of the capsid protein, has a ratio of 1 to 13,500. That is to say, you need 13,500 virus particles in order to get one infectious event. And we know that the infection is not cooperative. Each of those 13,000 particles has the same chance to start an infectious cycle. This comes out of the kinetics of infectivity versus the size of the inoculum. In other words, with a wild-type virus, if you put a certain number on a plate, you get these plugs here. With the debilitated virus, if you take the same number of virus on a plate, you get only 2 plugs or less. Which means the virus has, since the surface of the virus is not changed, because we didn't change any amino acids, the virus has problems as it gets in to initiate a translation, to the point where it can undo the mechanisms that the cell has in store to defend itself against the viral infection. And so once in a while, an inoculum will succeed, but the chances are, in this particular case, 1 in 13,000, which would mean the virus cannot be very contagious. You need a very large inoculum in order to infect a human. If you look at growth curves, not unexpectedly, if you look for plaque-forming units, it's the numbers of plaques that you form. The virus with more or less larger pieces of the segment of P1 will grow to very low titers. But if you don't look for titers, if you look actually to virus particles per cell, you see that the curves go almost to the same kind of yield, which means once the virus has induced an infectious cycle and has overcome the virus, the cell, it will produce as many particles as the wild-type virus. This is really very surprising, and this hasn't really been realized. There are lots of plaque-forming units being done, and it's been concluded the virus is very sick, it doesn't grow, and so on, when in fact the virus grows well, but it just doesn't make a lot of plaques. In order to find out what actually the real problem is with the virus, we cloned P1 again into the viral genome, but between P1 and P2, the replication proteins, we put as an indicator F luciferase. And in this disystronic virus, we had the R luciferase preceding the entire replicon. And then you translate that in HeLa cells in vivo, and you can normalize actually the amounts that you find by measuring for each transfection the R loop first and then the F loop. And what you find is, in fact, the poliovirus wild type, if we take it to 100%, indicates that the poliovirus with max translates just a little bit better. This is the one with overrepresented codons, the good ones with the good codons. Whereas those with various amounts, this is min, that still will translate a little bit, but not enough in order to overcome, in fact, the cell, and therefore not reaching the threshold of translation necessary to initiate an infectious cycle. And these are the two of the subclones, and they just reach, PB min just reaches apparently that threshold necessary. And that's the one with the minute plaques. Now, if you look, if you inoculate transgenic mice that are sensitive to poliovirus with these viruses, you find that the wild type you need in an intracerebral infection 10 to the 4 particles, whereas those viruses here with X, Y, or Z need 3 orders of particles more. Importantly, PB max was not more virulent in the mice than the wild type, and that was very important because we were worried that we may have actually produced a monster virus. And this is my last data slide, my second last data slide. You can immunize mice with these viruses here, which immunize by intramuscular inoculation, and then challenge with a lethal dose of wild-type virus, and the mice are all protected. Now, I will skip through the last 2 slides. We did actually look whether or not the CG content in our constructs is the one that governs the ability of the virus to replicate, or whether it is the codon pair bias score. And the answer is it's not the CG content. That's a bit of a surprise. And so codon pair bias is an interesting biological phenomenon operating in all known biological systems. The mechanism is poorly understood in our system. Translation is impaired. In the extreme case, below the necessary threshold for proliferation. Codon pair bias offers a new strategy for designing vaccine candidate. And we're actually very adamant about that, and we're begun to study some other viruses. This was done in Stony Brook on Long Island, and although unfortunately I do not have a picture with Steve Skina and Dimitri Pachav from the computer science department, the individuals here with the circle around were most involved in the work that I presented. Thank you very much.